Hi, my name is Andie Nelson, and my topic for today is animal testing. Just to give you a quick definition of what animal testing is, this is given by the Humane Society International. Animal testing is procedures performed on living animals for purposes of research into basic biology. They are used to test the effectiveness for consumer products such as medicines, cosmetics, food, and so on. So with that, my main claim today is that animal use in research and experimentation is ineffective, and I will be backing my proposition with 3 supporting claims. First, that animals are a poor comparison for humans. Second, that animal tests are extremely costly, and lastly, that better alternatives are becoming more available. Okay, so my first point is that animals are a poor comparison for humans. Animal tests are not a good source to reliably predict results in people. A study done by the FDA showed that 92% of drugs that enter into clinical trials after they have finished animal testing fail to be approved. So even if they passed in animals, they fail to be approved in humans in their trials. There are major differences in genetics, physiology, and biochemistry that result in widely different reactions between animals and humans. So an example would be the drug Vioxx, which was used to reduce pain and inflammation, specifically for arthritis. It showed good results in animal trials, and a report done by Dr. John J. Pippin showed that it had a protective effect on the hearts of mice, so it did help protect them. But once the drug went out into the public, it caused more than 27,000 heart attacks and cardiac deaths before it was pulled from the market and further discontinued. An inquiry done into this by the Physicians Committee for Responsible Medicine showed that the drug had caused heart problems in human clinical trials, but the companies used the evidence from the animal trials instead to get the drug into the market. There are also chemicals that are harmful to animals that are valuable by humans. An example would be aspirin, which is very toxic in animals, but is helpful in humans. Neurologist Dr. Aisha Akhtar is quoted as saying that even identical twins differ in their susceptibility to diseases and their response to medication. If we can't reliably apply one set of data from one identical twin to another, how can we expect to apply results from different species to that of humans? So there are numerous differences between animals and humans, and these species differences do not assure application between the species. My second supporting claim is that animal tests are costly. The United States Institute of Health spends $14 billion of its $31 billion annual budget on animal research alone. And a calculation done by the National Institute of Health showed that anywhere from $2 to $4 million is used for a single 2-species lifetime cancer study done on animals. A calculation compiled from the experiments carried out at the Charles River Laboratories in Massachusetts showed that an experiment involving gene exchange cost $22,000 in animals, while the in vitro method for the same experiment cost $8,000, so that's about half of what it costs to do in animals. And another experiment, which is DNA synthesis, costs $32,000 to perform in animals, and only $11,000 for the opposite in vitro test, which is a third of the cost. This leads me to my third and final supporting claim, that alternatives to animal testing are becoming more readily available. Animals have become the default method over time. Animal experimentation comes from a time when we didn't have the technology and resources that we do now. So the resources that would better serve our needs, we have resources now that would better serve our needs. Scientists have relied for decades on animal models to prove the effectiveness of treatments and medicines, and it does take a lot of time and persuasion to change these scientists' minds on what they're used to doing. It's like the old habits die hard thing. There's also a severe lack of global expertise and knowledge on this. Not many people know about the different methods, and many people are ignoring it in favor of the latter method. But these new methods do provide faster and more practical alternatives. One alternative is called Epiderm, which is an in vitro test made from human skin cells that has been shown to be more accurate in identifying skin irritants. In comparison studies done by the flagship company that made Epiderm, it correctly identified all of the test chemicals that irritate the human skin, whereas the same test done on rabbits misclassified 10 out of the 25 chemicals that were used. So the Epiderm is much more accurate in finding these. And there are also alternatives that provide results faster than that of the tests that animals have. The second alternative, which comes from the company In Vitro International, is called Corositex, which is another synthetic skin like Epiderm that is able to provide correct determination of corrosiveness on the skin materials in as little as 3 minutes and up to 4 hours. The exact same test when done on animals would take anywhere from 2 to 4 weeks, so that's a big time difference and a big gap that it leaves. Some animal tests take anywhere from weeks to months to even years to complete, and this alternative would provide faster results, resulting in quicker treatments and medicines going out to the public. So there are already 2 alternatives that provide more accurate and faster results than what is already being done. So in conclusion, animal use in research and experimentation is ineffective because animals offer poor comparison to humans, the tests are costly, and finally, faster and cheaper alternatives are becoming available. Thank you.
All right, the proposition, the secondary claims are labeled very clearly. It's easy to follow. Internally, you do good signposting on those particular points. Most of your supporting points include an explanation and some additional definition. There's usually some information that is cited on those particular points. I'm not sure that the examples are sufficient to make the claim that you're making, but I thought that the examples were actually pretty solid and that you usually describe them in good detail, in particular the last 2 when you're talking about the skin irritants and those alternatives. There are some underlying issues that jump into my head immediately. If these other tests are half the cost and they're more effective, then why researchers wouldn't be using them is a little unclear. The only explanation I get for that is that, well, there aren't a lot of people who have expertise in those particular fields. But that's the one thing that seems to be not very well cited on this. That's a presupposition, and you need to have some data on that. It would seem that we got some information on the totality of the number of medications that have been approved and then later turned down for human use after they were approved on animal use. What would be helpful is if we had a total number of medications or applications that came so that we could tell whether or not the 2 or 3 cases that you cite represent a substantial amount. Yeah, I mean, you mentioned Vioxx, for instance. That's definitely a dramatic example with the number of deaths that have been associated with it. However, if there have been 10,000 products that were submitted for approval, but only 8% of them, as you say, ultimately were correct, that still represents 800 medications that are now available that weren't available before, and you've got one example that seems to suggest that there was a problem. So I'm not like I said, if we're making inferences here and we're looking at all those numbers, they need to go together a little bit more effectively. I thought you did a good job citing the sources of all your information as well. There's a little more reading than you want, and of course you're trying to get it done quickly, so maybe the pace could be a little bit more reasonable. All right, thank you. What's our time? Yeah, we got to stop here. Nathaniel, we're going to have to hear you on Monday.
Okay.
Sorry about that.